Hello everyone, my name's Zach and today we're going to be talking about synapsis. So what is synapsis and crossing over? Synapsis is the pairing of homologous chromosomes during meiosis. It's where they line up. And so crossing over is the sharing of genetic material between two different chromosomes. So excuse my art skills down here, but I drew out a little demonstration here. So on the left, you see the two chromosomes here in the center are lining up. In the middle, the actual crossing over event is happening, so those arms are crossing over. And then over here to the rightmost, you see that genetic material has been exchanged. And so we're going to go into some examples of that. So here in the first individual, you see the original chromosomes up here in the top left. And so you can see that there are regions that match up, the M and the L and the H and the G. You just have to flip the second one around. But you can see that the center sections, K, J, centromere I, do not line up. So to compensate for this, the chromosomes will actually form this omega in this loop to match up as many regions as possible. And so you can see the 4 gametes that are produced right down here, and those are actually color-coded. So all you got to do is follow along the colored paths, and you'll see how these gametes are produced. The red and the blue are actually the same as the parents because the loop doesn't affect them. But the center two, you can see the green comes up here, but then it crosses over, it goes around, and then the brown does the same thing over here. So now we're going to talk about viability of these gametes. So for the gamete to be viable, it needs a centromere as well as it needs all letters. And so you can see the top 2 are viable because nothing happens to them, but the bottom 2 are not because they both have a centromere, but they don't all have letters, all the letters. So this bottom one doesn't have an H and a G, or this green one, and then the bottom one, which is the brown one, does not have an M or an L, so those 2 are not viable. So for the next individual, you can see the parent chromosomes up here in the left-hand corner again, and all you have to do is flip the bottom one in this case, and it is the exact same as the top one, but there is a crossing over that occurs in J and K. Now, the thing is here is that since they're all the same and they line up, really all of these letters, you're still going to get all letters and you're still going to get a centromere because they line up just right. So you can see here that all 4 of their gametes are viable because they all have a centromere and they all have all of the letters. So this individual has a better chance of surviving because it has more viable gametes, or a better chance of passing down its genes. Alrighty, and so here are my citations.